Okay. Welcome to our distinguished lecture series today. This is a lecture series where we invite our thought leaders internationally from all over the world. And we are very happy to also welcome to the people online. And we are very happy to welcome Professor Halina Rubinsztein-Dunlop from the University of Queensland today. She's a professor of physics at UQ. And she is also a Deputy Director of Center of Excellence in Quantum Biotechnology and was also the Director of Translational Research Program at the ARC of Engineered Quantum Systems in Australia. At UQ, she leads several research groups on quantum atom optics, laser micromanipulation, and biophotonics. She's the recipient of many national and international awards. For instance, the officer in the General Division of the Order of Australia, a Fellow of the Australian Academy of Science, a Fellow of SPIE, Optica and AIP. And she was recently awarded the SPIE Directors Medal in 2024. And also this year, the SPIE Gold Medal 2025. She is also maybe interesting, so several of us actively pursuing work in equity, diversity, and inclusion. And she promised to also take part at a summer party later on. So if you want, you can talk to her later. And with that, I want to welcome you on the stage. And the title of her talk is Sculpted Light in Nano and Microsystems in Quantum Biotechnology.
Thank you very much. Thank you very much, Sylvia, for this incredible introduction, and also for inviting me here. Whoa, this is very loud. Can we put it down a bit? Thanks. And for me to be here and be able to talk to you about sculpted light and in nano and microsystems, and then on a special request about quantum biotechnology that Sylvia wanted to listen to it as well. So, as Sylvia said, I come from the University of Queensland, so that's this one here. And I'm part of two centers of excellence, the one which ended in May 2025 in Engineered Quantum Systems, and now I'm Deputy Director of the Center in Quantum Biotechnology. So this is the system organized by Australian Research Council, where we are funded for 7 years in a row, to pursue research which is more than individual grants for basic research. And it's supposed to lead to applications. And in all of those centers, there is more than one university involved. So in our center, in quantum biotechnology, we have five universities around Australia, and we have partners both from international universities around the world and from industry. Okay, so I want to show you a little bit of tricks that we can do with light. So we like to play with light. So what you are seeing here are tiny particles of the order of 2 microns in size, which are rotating in orbital angular momentum light beams. And we can create any light beams we want. So here, for example, we create Bose and Einstein in Bose-Einstein condensate. So what you are seeing here is basically we have taken and created light in Bose-Einstein images and put our Bose-Einstein condensate into it. And and we play with light. And this is just to show you how clever we are. Anyway, and I wanted to show you another one. Okay, and this one here, no, it doesn't want to play. Let's see whether it will play here. Okay, so here it plays. So you see OMG, and Vahid said, oh, that's nice, you have OMG. But it's not, oh my God, it's optical micromanipulation group, and this is actually done with holographic tweezers. So that many tiny holographic tweezers, each one tracks the particle, puts it in the pattern, and we play with them. For what? For nothing, just for you to laugh a little bit about it. Okay, so back to work. So what I want to talk about is how I can use structured light, both in nano and microsystems, And I want to give you examples of it in quantum enhanced imaging, and talk a little bit about quantum interdisciplinary research, and also go back to structured light again and give you examples of quantum atom optics where we use structured light, and motivate why we are using structured light and how we move from pure research using this structured light to how we use quantum atom optics in medicine, which is part of our Center of Excellence in Quantum Biotechnology. And then my beloved optical micromanipulation, the same thing here. I want to use structured light to look at light-matter interaction and use the structured beams carrying orbital angular momentum for transfer of angular momentum of light to matter and building micromachines, etc., etc. Okay, so that's what we want to do. And this UQOMG, that's written with light using spatial light modulator. So you can see that you can vary the intensity of light and you can make light into any shapes you want. Okay, so back to work. So we all know about first quantum revolution, and it was a big thing in the beginning of 20th century when these big people in the pictures here came up with wave-particle duality. And the statement by Niels Bohr is really saying, if quantum mechanics hasn't profoundly shocked you, you haven't understood it as yet. And it led to incredible development in our lives. It led to things like semiconductor industry, which has developed very quickly, MRI, and then bringing the astronomical things into the lab and being able to look at it in the lab and probe it. And then we are now in the second quantum revolution, where Richard Feynman, very long time ago in 1982, said, nature isn't classical, damn it. And if you want to make a simulation of nature, you'd better make it quantum mechanical. And then things started to move. So we see where second quantum revolution took place, and then it's moving, and it's achieving incredible things. And many of those incredible things are done in this institute here, and I was witnessing many labs doing fabulous things today. And so if we look at the second quantum revolution, what it brings us is that we go from understanding of quantum mechanics to engineering and trying to find applications which weren't possible before. And so we go from cart driven by horse. This is not Tesla, okay? This is Polestar, okay? So it's fully electrical car, but not Elon Musk stuff. So we are saying by engineering quantum technology, it is possible to achieve capabilities far beyond what was possible today. And we are moving forward very, very quickly. And so we created the Center of Excellence. So I call it Center of Excellence. It sounds very profound, but what it means really is that Australia decided to put money into research, into endeavors that would cover, as I said, bigger endeavor in science. And so combining many universities together, which would go towards common goal and not work in silos about different parts of the program, but always in big collaborations. And big collaborations between these 5-6 universities within Australia and with partners all over the world and industry. And so our mission is to harness quantum technologies for the life sciences. It's not a small mission. We are pretty ambitious. And we want to lead to a global transformation with impact across energy, health, and agriculture. So ambition is there, okay. And so we have 3 programs. We call it molecules, cells, and brain. And I will give you a little bit of go through them. So we know about the development in quantum computing, and we know that we can do quantum sensing and imaging. And actually our center stems from another Center of Excellence, which was in existence for 14 years, which was in engineered quantum systems. And we were there developing quantum sensing and imaging as well. And so we're now bringing those results into bearing in biology. And so we want to close the gap in imaging and modeling. So if we look at this figure here, an imaging, we can look at what electron microscopy can achieve. So we are looking at the size versus time scale. And if we want to look at real biological systems, which can be dynamic, we have to reach these scales here. And that's quite difficult because, as I said, electron microscopy is here. It's fantastic. The resolution is down to single atom. And light microscopy, we also have fantastic super resolution microscopy with Nobel Prizes there and more development taking place. But can we do better so that we reach this sort of imaging here of biological processes? If we go to modeling, so we can model biological, again, time versus size, and we want to do molecular dynamics or quantum chemistry. We want to go to the quantum, which will be in femtoseconds, and of the order of single molecules. So what we want to do in imaging is to do quantum imaging to push it towards the time scales and sizes that are needed for helping imaging in that way. And the same goes for modeling, that we want to reach the quantum computing, using quantum computing and quantum algorithms, reach this region here where we can do modeling in proper way. So the quantum solution is, again, quantum chemistry and going to MRI in those regions. So as I said, we have 3 programs. One is in molecules, and that program, the goal is to image and predictively model the structure and real-time dynamics of single protein at room temperature. So I have seen fantastic things here in this sort of area today, and we have a little bit more ambitions. Whether we reach there or not is a different story, but that's what we want to do. So basically, we can look at things in using different techniques, but the best thing we want to reach is the single molecule detection and the modeling of them, and single molecule detection in room temperature. And then our second program is in cells, and this is discovery how cell scale behaviors emerges from molecular scale interaction. And so we want to go to this intracellular processes and being able to image them, model them, and understand them, and answer very complicated biological questions. And then we want to go to brain, and we want to achieve real-time imaging of whole brain electromagnetic fields at subcellular resolution. Again, a lot has happened in this area, and a lot is happening around the world in this area. And there are fabulous devices today, like squids, which work very well in this area. But these are not room temperature devices, and they're a little bit difficult to use. Now, and we want to do it in the device in room temperature and develop them in that way. And then we want to go from single neuron to organelles, organoids, and full brain, and also modeling of it as well. And so another example is magnetic encephalography. I have difficulty pronouncing it. And this is an existing tool today, and it's used. And there are about 100 machines operating worldwide. They're incredibly expensive, and they're difficult to put our hands on. So wouldn't it be fantastic to have magnetoencephalography done in the way which is done in room temperature, and with the devices that are small enough to be put in general practitioner's office and be able to be used there. Okay, so a little bit more about brain. So as I said, we want to achieve real-time imaging of whole brain electromagnetic fields at subcellular level. And we want to use imaging, we want to use optomechanics, and here is optical tweezers that we have done in looking at the brain of zebrafish, neuronal networks in zebrafish. So we want to combine all these methods and go further to looking at, as I say, organelles, organoids, and full brain. Here we were lucky enough to be able to look at the full brain of the zebrafish. And if you ask why zebrafish, well, because it's mammalian, model of mammalian brain, but it's small, so it's easier to study. And we were able to use optically based methods, so biophotonics-based method, to investigate the whole brain by cheating the fish that it's moving. And if you're interested in it, I won't talk about it anymore, but I can tell you plenty about it later. Okay, so that's that. So this is what we want to do in quantum. So one example of quantum-enhanced nonlinear microscopy that we have done in the center, it's quite old work and predates the center in itself, but the group is developing it further. It was done under Professor Warwick Bowen's group. And that's basically looking at Raman spectroscopy done with squeezed light. And that gives us imaging which is improved without increasing light intensity, but using this quantum photon correlations. So you have squeezer here added to the system, and you look at the correlation, and you get enhancement of the Raman signal, which is quite substantial. And if we look a little bit more in details on that, so you are here reaching under shot noise limit. It's improvement. We would call it big. It's not really huge, but it is definitely improvement. So you are using this Raman spectroscopy, which we call quantum Raman spectroscopy, which is below shot noise limit. And if you look at the quantum enhanced SNR limit, it's a little bit better, and it's, as I said, below shot noise. And also, and to show you the examples of it, here is the imaging. These are the little spheres of the order of 3 microns in size, and you can image them very well. And then here we go to a cell and look at the organelles of the cell, and you can recognize them very easily, and the enhancement is taking place here. And you can see what's happening around the membrane as well. And in this paper, they have been able to show that the damage made by the intensity of the light is minimal or almost non-existing. So there was one.
What kind of ramen was that again?
This is the stimulated. Oops.
Oh, okay, stimulated.
Yeah.
And the squeezed light was stimulating.
That is your squeezed light. Oops. Sorry. There is the squeezed light going in. So that's your squeezer, and which is then put in the microscope, and you're looking at the correlation.
Okay.
And it gives improvement of 3 times or something like that. So it's quite substantial. It's not absolutely fantastic, but it's on the way, and it's definitely under shot noise limit. Okay, and most importantly, it does not destroy the system. So it was also looked at in the division, under the division of yeast cells, and yeast cells survived. And it's being developed further. So now to my structured light, which is my dream research part. Okay, so structured light, there are many ways in which we can construct structured light, and in your labs here I have done it with the Marucci plates and also with SLMs. So we use either SLMs or digital micromirror devices, and it's a little bit much of a muchness. It depends what you need from them. So if you need dynamical range, you will use digital micromirror device. And if you don't need dynamical range, you use SLM with liquid crystals. So basically, they perform pretty similar roles. You can vary intensity and phase, And depending how much money you want to spend on them, they can be better or worse. But they do what they're supposed to do. And so digital micromirror device, I haven't seen it here, but I'm sure that you have it here somewhere. And it's basically the device that you have in those projectors here that you are looking at my slides with. And there is a collection of tiny, tiny mirrors which can be wiggled with certain angle, which is about 19 degrees. And I can program it, how it wiggles, and I can then program my light, how it is scattered or transmitted through the device, and have certain resolution. So we have used this digital micromirror device for looking at BECs, and did a lot of research with that. But to show you how clever we are also doing a little bit of art with a structured light. So what you are seeing here looks pretty grainy, but this is Mona Lisa written with digital micromirror device in Bose-Einstein condensate. And although it's grainy, you can see her eyes pretty closely, quite prominent eyes there. They are about 1 micron in size. So it's pretty good resolution. It's about half a micron resolution. And of course, if you come from Queensland, you have to have this Queensland fish with colors. And then, of course, we have to advertise ourselves. So these are our faces in Bose-Einstein condensate with DMD structures. And we can do it better. We can use feed-forward techniques, algorithms, to improve this Mona Lisa image or any of the images we have, because a lot of things that we want to do with very well-constructed light, we need the distribution of intensity to be very predictable. So we go from no feedback control with 4-tone Floyd dithered feedback control, and you can see that the improvement is quite substantial.
So can you see how you make these pictures? I mean, the thing is with the Bose-Einstein condensate that every atom is delocalized to your 100 micron or whatever this was, and we have...
So the atoms are not in her face, but in the surrounding. So they are pushed, it depends on the wavelength of light, so they are pushed away. They are detuned, and so they go to the places where the field is not.
So why don't they go to the... I mean, the Bose-Einstein condensate is a delocalized...
So Bose-Einstein condensate is a cloud, okay? Then you put the pattern onto it. So first of all, I will show you that. We construct it in a 2D fashion, and then we put this pattern on it. Okay, and depending on what sort of wavelength of the detuning you have, to red or to blue, you can get the atoms going either in or out. Okay, so what do we do with these lights? We do a lot of things with them in basic research. So this is my UQ Bose-Einstein condensate lab, and so we have these atoms in first in 3D MOT, and then it goes into our cell where we construct our Bose-Einstein condensate. And then we take our structured light, and we can structure it in being in rings. So this is binary image, projected image. This is what you were asking about, and atomic density, okay? And then, as I said, we can do Bose-Einstein or whatever we want, and fields in a ring or whatever. And we can, with feedback control, have them very evenly distributed intensity-wise and measure different things with them. So I don't have to explain to you how you get to Bose-Einstein condensate. You all know that. But this is the principle here. And then we use it for studying quantum turbulence and other funky things. And I will show you one example of that sort of studies. Where doesn't it want to go? Okay, so just summarizing again what we do. So we do turbulence and sensing. So we look, for example, how the yellow dot on Jupiter, a giant yellow dot on Jupiter, how it is functioning and what it depends upon, and we can study it in the lab. And then I will show you an example of this. And then we look at the atomtronic circuits, which I will show you, and then we are doing other things like quantum simulators with our cold atoms, and we put our atoms into the rings, Bose-Einstein condensate into the ring, and we're looking at measuring magnetic fields, very, very precisely, rotational dynamics of the fields. Okay, so superfluid atomic circuits. So as I said, we have our Bose-Einstein condensate here, and I come with the sheet of light, which puts my Bose-Einstein condensate into a flat sheet, and then I come with my DMD pattern, and I put it into the pattern I want them to be in. And so in this example of superfluid atomtronic circuits, what we do, we have dumbbell construction of our light, and we can start our atoms in one of the dumbbells, in the lower one, for example, and then turn on the channel in between them and move them up and down and get them circulating back and forth. So this is a simple atomtronic circuit, so superfluid oscillates between the two reservoirs under initial chemical potential bias. Okay, and depending on the bias, it will behave differently. So you can see the modeling of the system. So I have 11% initial bias. I start my atoms here, and they go back and forth. And why did it stop? I don't know. If I go back. Okay, so you can see how it's oscillate back and forth, and this should be going as well. And here we go. Under higher bias, it all happens much faster, and you can see that the signal here, the Josephson dynamics is very nicely here depicted, and then when the bias is much bigger, it sort of disappears very quickly, and we can calculate what is happening with it. These are vortices being built in the system, and we study those vortices as well.
Those are light.
Mm-hmm. Mm-hmm. Okay, so this is real life stuff. So this is the experiment, and the good thing about it is that, of course, this is collection of images, okay, and the reproducibility of each time of the system is good enough to then put those images together and have the film that I'm showing you here. So basically, I start with the atoms in this lower dumbbell, and then I move them up. And I can vary here in dynamical way. I can vary the length of this channel. I can vary also the width of the channel and see how it oscillates back and forth. And then I can analyze it all, and I can analyze it as acoustic model for an atomtronic oscillator. So basically, this is my acoustic Helmholtz oscillator, and then this is what we have, this LC circuit is presented here. And you can see that the final result of resulting frequency is dependent upon the density of the atoms in the reservoirs, but it's also dependent upon the length of the channel and the width of the channel. Okay, and we can very quickly analyze that under different conditions. So this is just one example of how we can do Bose-Einstein condensate investigations and build atomtronic circuits. And as I said, we do turbulence, we do quantum thermodynamics and all sorts of stuff. But we also are thinking about applications. Okay, so here's one application that we haven't done, but we thought of and are thinking of and putting together. So basically what we want to do with our atoms, not necessarily Bose-Einstein condensate atoms, but Rydberg atoms, and they can be in room temperature, is to have non-contact ECG machine. So that's a vision, okay? We got funded for it, but we don't have the equipment yet. Okay, so what is ECG? Everybody knows, right? I suppose many of us have had it before. So you have electrodes, you put it on the body, and you have electrocardiogram, and you look at ECG signal, and if it looks as clean as it is, the person is healthy. Okay, but wouldn't it be fantastic if you would have this ECG machine away from the body if you wouldn't need the electrodes. The question can be asked, why are electrodes not good? So we actually got funded, would you believe, from the source of the money from our state government for Olympics, Quantum Olympics 2032. How does that sound? Like science fiction, right? So we applied for the money to monitor the muscle function and heart function of sportsmen, okay, in function. So they are running, and you want to do ECG on them. And of course, that's very difficult. Even if it's on the stationary bike, it's difficult to put the electrodes on. And you want to train them, you want to show them how to run and how to use the muscles, and you want to measure the signal, and the signal is super weak, etc. So can you have an instrument that is away from the body and is looking at the function of the heart? Now, that sounds like science fiction, but it's actually an actual problem, okay? Why for the Olympics 2032? Well, that's a good question, because we want to train our sportsmen, and you know, if you know anything about Australia, you know that sports are very important. Not that I agree with it, but that's the way it is. So anyway, but it's also very important because it's very important in, for example, neonatal care, when the baby is born premature and you want to follow its heart. The skin is rather fragile, very thin, and putting electrodes on the skin is not very nice for the baby. And so if you would have an instrument that is put away from the body, that would be very helpful. Now, if we want to take such instruments, so here, for doing all these measurements, you need a specialist. Wouldn't it be nice if it would be 10 key operation stuff that you can put in remote areas? You know, in Australia, we have a lot of remote areas around us. And so having healthcare in those areas is very difficult. So having a device like that, where you turn it on, and it gives you a fantastic result, would be a great thing. And then it's also done in tropical places. And so putting electrodes on the body in tropical places is a little bit difficult because of sweat. So you can, of course, wipe away the sweat, etc. But the climate is not very nice. So if you would have something like that, it would be very helpful. So there are real reasons, apart from sports, to do it, although sports is most important. So advantages, in summary: no skin irritation, and don't have to shave people. Calibration-free, we hope it is. And it's possible that it can work in 3 dimensions, and no need for the expert. So as I said, it's non-existing piece of equipment, but that's what we're dreaming about. So this is how the standard instrument is working today. And the drawbacks of that are given here. I won't read them, but believe me, they are. And there are many reasons why heart diseases in Australia are very important, that they are important all over the world. But if you go for the money in Australia, you have to have good reasons for Australia. So here we have them. Anyway, so what we are proposing is that we want to do it with Rydberg atoms to sense the bioelectrical fields of interest. If so, can they do so under realistic condition inside the glass cell? So I have my rubidium vapor in a cell, okay, and I want to sense the electrical field with it. And the question is, can this measurement be performed in clinical environment? How do biological tissue affect the signal of interest? So if I put my device a little bit further away from the body, what is the skin doing, tissue doing to it? How is the signal diminishing, etc., etc.? And then look at the needs of Aboriginal communities and Torres Strait Islanders communities. So here's the proposal. This is nothing that we started to do. There is existing measurement in this paper from 2023 by Ling, and that's electromagnetically induced transparency, and you can measure fields with it, and the sensitivity is pretty good here. And so basically, this is the scheme that we want to do. So you want to now have vapor in the cell, and then do this electromagnetically induced transparency. So this is the system that you have. Here is my cell with the electrodes, and I can put different fields on it, and I can measure the electromagnetically induced transparency here. But the question is, how well can I do it, and what are the problems? So, as I said, those experiments have been made, and this sensitivity has been achieved. But the question is whether A, we can repeat it, B, can we beat this sensitivity? And our approach for that is that we want to put it in optical cavity and enhance the signal doing it in an optical cavity and go to much lower sensitivities. Now, there are problems, and we have heard a little bit in one of the groups today what are the problems with rubidium, rubidium in fibers, how the rubidium deposits itself in the fiber. So the same thing happens here. So if I have alkali metal in a vapor cell, and if it's normal cell made out of glass, normal glass, then that it covers the glass. And no lasers will come through, and no electric field penetrates the cell. So we have to construct the cell that is from some other material, and I was asking around whether you do anything in quartz, but didn't get an answer yet, so we can discuss it a little bit later. So again, so this is the proposed arrangement that we want to have and want to achieve the sensitivity of minimum field measurements of the sort that is depicted in this figure. So this was all dream. At the moment, we have all the lasers needed for the experiment, and we have normal cells, rubidium cells. So normal glass cells with rubidium you can buy everywhere. Thor Labs sell them. People sell them. We used to make them in olden days, but those days are gone, and we have never made them from quartz. So if you know anybody who makes them in quartz, please let me know. I would be very happy. Anyway, switching fields a little bit. I want to go now to structured light in optical tweezers in optical micromanipulation. So what I want to do in optical trapping is to go to below quantum limit. Am I there yet? No, that's another dream. So we have a lot of dreams, but anyway. So normally what you do in optical tweezers, you're looking at the thermal motion. And so if I have free diffusion of a particle, so this is how free diffusion will look like. If I trap the particle, you can see that I confine it in optical potential, and it still wiggles with Brownian motion, which I can measure and use for my measurements, but it's much more constrained in movement. And this is the modeling of how it all happens. So I have particle going into highly focused laser field, and if I'm lucky enough to be able to detect all the transmitted light, I can look at the properties of this transmitted light, and that gives me measure of position, force, torques, and whatever is acting in the system, down to angstrom in position, femto, Newton in fields, and equally low in the torque. So this is modeling, but that's what we're doing in practice. Okay, so this is how it looks like, and I don't have to show it to people here. You do trapping here as well, but I will just mention it anyway. So you see here 2-micron size particle, which is trapped in optical tweezers, and I move it in X and Y. I can move it in Z as well. And you can see that the particles which are not trapped wiggle with Brownian motion when they are free. This one wiggles as well, but it wiggles much less. And so if I model it, I can model it as simple harmonic oscillator, and then I can plot mean square displacement and function of time. So you see that was what I was showing you before. now I have the spin squared displacement, I can analyze it, and I can look at power, spectral density, and then it gives me characteristics of the trap. So I can look at the trap stiffness, which is dependent upon the particle size and viscosity of the liquid in which the particle is. And I can do the measurements. And that was all based on transfer of linear momentum of light. But we can also transfer angular momentum of light, either in the form of spin angular momentum or orbital angular momentum. And so, again, in one of the labs, I saw orbital angular momentum, LG01. So here we are using LG03. So basically what it is Gauss-Laguerre beam of light, where per photon I carry plus minus LH bar per photon of orbital angular momentum. These are LG beams are like donuts, but where the pointing vector is describing this sort of movement here. And so we transfer, you can see here, I'm putting dumbbell particles into that beam, and I'm rotating them, and I can rotate them very fast. And this sort of things have been rotated up to megahertz. Okay, I can transfer spin angular momentum, and per photon it will be plus minus h bar per photon. And here is calcite crystal and vaterite crystal rotated in spin angular momentum. So this is about 3 microns, and this is about 3 microns. And we want to deal with this one because it's spherical and therefore much easier to look at, to model, and we can use it for viscoelastic measurements of biological systems. And this is, I would point out, that this is still very simple Gaussian beam, but which is carrying plus minus 1 h bar per photon of angular momentum. So this is optical tweezers in transfer of angular momentum of light. Okay, so this is how it functions. So I can use this transfer of spin angular momentum for measurement of viscosity. And the way it works is that my probe is like lambda over 4 plate. Okay, so it's birefringent plate, well, sphere in this case. And I'm coming with the light, which is perfectly circularly polarized, and I transfer, so I have now torque acting on it, which has to do with the change of the polarization when the light is transmitted through the particle times the power of the laser that I'm putting on it divided by the frequency of the laser. And I can do this measurement. So if I'm collecting all of the light, I can measure this torque, and that can be then showing how I can determine viscosity from that. And so basically 2 types of measurements can be done on this. We call it vaterite particle. It's calcium carbonate in form of vaterite, so it's birefringent particle, and you can either rotate it in circularly polarized light, or you can align it certain angle, as you can see here, and then look at the viscosity and elasticity of the system using it. Okay. And so, what do we do with the system? We use... What determines the speed? The speed is determined basically by the birefringence, the difference between 2 refractive indices, and also the power of the laser used, and the size of the particle, and viscosity. Okay? Okay. And we can spin them up to megahertz. Okay. Eventually they disappear. So people have done those measurements when you transfer spin angular momentum to the particles. They have done it in vacuum as well on our vaterite particles. That's Kishan Dolakia's work. Okay, so what can we do with it? We wanted to go to measurements. I saw beautiful examples of raffles being built in one of the labs using your iSCAD, right? So here it's not done with iSCAD. This image where we're cheating the cell to eat vaterites is done with fluorescence microscopy. But anyway, so the process we want to study is macropinocytosis. So that's the process, everybody I think knows it, but I will repeat it anyway. So the cell is building raffles. Will that play as well? Because the raffle is quite visible here. No, it doesn't play. This should play. Okay, so cell is building ruffles, and we pulled our vaterites in the vicinity, and the ruffles close on themselves, and they go into the cell in this sac here. And the idea is to get our vaterites particle inside this macropinosome, and then it dynamically shrinks and delivers the nutrition to the cell, and the maturation process takes place. And this process is very different for the cells which are cancer cells versus healthy cells. And that process is not that well known. So determining viscoelastic properties of the systems in dynamical way on very short time scales is very important. And so that's what we're doing here. So we're cheating the cell to eat our vaterite. It eats it. It's in the cell. And now I apply my rotating tweezers. And this is the process which takes place. So the particle goes in, and then I apply the tweezers here, and I look at the rotation, and I can show you in the next slide that I can then plot and see how the viscosity is taking place, changing dramatically in the cell. So here is the rotating particle inside the macrophage in the macropinosome.
How large is the particle? How large is the particle?
1 micron. We want to do them with much smaller particles, but we haven't done it yet. Okay, and the smaller the better, but then as the particle gets smaller, it's a little bit more difficult to trap it and rotate it. So there is some golden rule there which we have to follow. But now we want to do it on very, very short time scales, okay, dynamically. So what we are interested in is looking at these processes in dynamical way on very short time scales. So again, in this paper here, which comes from Warwick Bowen's group as well, what they have done is to look at passive motion of the optically trapped microsphere, looking at the velocity instead of looking at the displacement position. So basically, this is what is measured normally in optical tweezers. That was what I was showing you before. You measure the changes in the position as the particle is moving in this potential that you build in optical tweezers. You look at the position changes. But if you instead look at the velocity changes, the curve is changing. So this is the normal curve that we have seen before. And you can look at the changes in the viscosity here. And then if you instead look at the velocity power spectrum, then you have these two frequencies. And at very high frequencies, you can, in very short periods of time, you can measure the velocity power spectrum. And we have done it in rotational geometry. So basically, here you have Langevin equation, where you have rotational inertia, you have optical torque, and you have drag torque, and a normally distributed thermal torque. And normally what you do in optical tweezers is in rotational optical tweezers is that you say that the rotational inertia is so small that you put it to 0. But if you can measure it, that's what you will be able to do. So, so the position power spectrum looks like this, but if you do the velocity dissipation, it will give you this equation here. And so you can now determine the inertia in the system. And so this is the measurement of the angular velocity. So that's a real measurement in the system. And you look at the angular velocity, and then you look at it in comparison with other measurements, and you can see that it's slightly better than, and you can measure that inertia in a very short period of time. And so there is a hope that if you can measure these two frequencies now, you go to very high frequencies and therefore very short periods of time of measurement, and you measure dynamical system in very nice way. Okay, and to finish it off, I just show you this curve. And so what it basically shows me here is that I have this decoupling from the translational component, and I can measure high bandwidth, do high bandwidth measurement and reach the ballistic regime. And we have done that. We have not applied it to any biological system yet, but the hope is that I can do it very soon. Well, I can't, but Mark will be able to do it. And so with that, there were just few examples. We want to reach, as I said in the beginning, we want to reach quantum limit. We haven't done it either. Whether we use squeezed light for that or what we use, I'm not quite sure. I'm thinking about it. But the plan is to do ballistic regime at quantum limit. And I want to use it in real cells, living cells. And I also want to look at the protein condensates and use the quantum light in imaging. And of course, my ECG with Rydberg atoms. And this is the quantum atom optics group, so we work closely with theoreticians, Matthew Davis and Agravan Safavi-Naini, and there are students and postdocs, and my colleague who is running the lab with me, Tyler Neely, and this is the micromanipulation group. And thank you for your attention.
A lot, Halina, for this very inspiring talk. We can have several questions. I give you the...
Oh, that's me. Sorry. Yes, it is. Yes, it is. I was showing how the particle can dance. Sorry.
Thanks very much, Halina. I have a question to start for your Rydberg atom electric field measurement from the heart. First of all, we can probably help you with the quartz cells, but
Sorry?
we can probably help you with the quartz cells.
Oh,
but,
fabulous.
the electric, you showed that you expect some like 50 nanovolt per centimeter. That's like the sensitivity that you have from, that's the electric field that you have of the heart at the position of it. Now, these Rydberg states are very sensitive to electric fields. That's why you want to use them. But the walls of glass will also have stray electric fields. Have you thought about, or have you measured how much that is and whether that's going to...
No, we haven't measured anything yet. And sorry, we are very much aware of those problems, and that are the first measurements we want to do, what the walls are doing. But I think that the bigger problem at the moment is the deposition of the rubidium on the walls, which will stop everything, right? And then...
I want to have a little heater there, and then you...
Yes and no. You can have a heater, you can have blue laser, which will also put the vapor away, but if you want to do it in the cavity, eventually we want that cell to be a cavity as well. So if it deposits itself there, that wouldn't be very good. But all those problems are the problems we have to solve.
All right, thanks.
But of course, if I have your quartz cell, then I'm happy.
For the very nice talk. I have a question regarding one of the very first slides that you were showing the direction of the center. And you mentioned that you aim for the full imaging of the brain at the single neuron resolution. If you elaborate, I mean, if it is possible, I don't know if it's a secret.
So I can elaborate, but we don't have it, right?
But what is the approach that, is it FNIR or what is the approach that you would like?
The approach is to go to imaging it with nanodiamonds and NV centers in diamonds, microscopy. And how it will be done, I don't know. On single neuron level, it's done and it's working very well. In organelles, it is also starting to be done. But how you move from there to full brain beats me at the moment. But that's the aim of the game. Sorry.
My question is about these rotating particles with the optical tweezers. You said that the rotation speed depends on the birefringence. What if you make the particle chiral? What if this particle is chiral? Will it...
It will rotate as well, and people have done that. You can measure chirality using this as well. But what I would like to have is a birefringent material which has the two refractive indices with bigger difference between them.
But it's hard to make a particle out of such material. For instance, a crystal. Is it easy to make a perfectly round particle out of, let's say, calcite?
Yes. So the reason why we have birefringent particles, which are spheres, is because doing calculations and computer simulations on spheres is much easier than doing it on arbitrary shaped particles. However, we have optical tool, and somebody was telling me that they successfully use our optical tool here in this building, where we can calculate forces, torques, and displacements for arbitrary shaped particles. And that means that you give me a crystal, if I can describe it, right, then I can use DDA or other methods for describing how to interpret my signal. Have I done it? No, but could do it. And there are particles which can be produced, but they're very large, and they're magnetic, that have very large difference between two refractive indices of about 0.5, right, which is huge. And so it would be nice to have those, and magnetic particles are quite attractive because you can measure much larger forces. No? 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 Okay, I won't try. Okay. Retract it. Okay, sorry.
We suffered from that.
Okay, I will stay put, yeah. Yeah, but it would be nice if you have particles like that, we would do them, yeah.
So staying on that topic, I think measuring local viscosity is a very important issue in the cell, and that looks really fascinating, but I'm asking whether you end up having issues because you're really kind of like putting a cappuccino foam maker into your thing and spinning it around.
No, I do not actually. So viscosity can also be measured with moving particle in X direction.
Or just looking at the natural diffusion of the particle.
You can, but with the rotation, we can show that Faxen correction is not necessary. So the influence of the environment is much smaller than it is in translational case, not in the case that you are describing, but in the natural case. So for example, if you want to look at the viscosity at very narrow channels, then doing translational case it gives very big influence on Faxen correction.
I was just worried about the friction that you create
There
and
is
the
no
vortex that you create in the cytoplasm, kind of moving things around.
Look, well, the other problem is whether I can spin it in the cell. I mean, in macropinosome, I can spin it because it's full of liquid, right? And it's much bigger than my cell. But if I'm in the cell and try to spin it, and I'm close to...
I thought you showed that you did that. That was
yeah, but it was in macropinosome. It's still in macropinosome. We did spin them in the cell, but it is a big problem there. You are absolutely right, because the cell is tight. Yeah. There are a lot of things happening there. So you either find the place... So I can take normal tweezers, traditional linear tweezers, linear momentum transfer tweezers, and if they're strong enough, I can move them around the cell. I can do that. I can spin them also, but whether I create those craters is a different story. There is a question far away. At the...
No, you need this.
Hi,
Thank you for the amazing presentation. Maybe I have a more trivial question. I was concerned about the BECs, and if I understood correctly, then you first make a BEC in a 2D plane, and then you shine your DMD pattern on it, and then you coagulate in some places and you release the atoms in others?
No. What I do, I create them in BEC, the pattern, and then I let it go.
I see.
Okay.
You release it in space.
Release in the space, and I measure it.
Okay, that's very interesting. And the second question is how you color code the images that were color coded, because if I understand this, the only information you have is the scattered light.
Yes, it's fake color coding.
I see.
How does that sound?
One more question, maybe.
Yeah, sure.
Why Rydberg atoms? Why not lithium-6 or, I don't know, other atoms that you could use? What is the special reason for
So
using
Rydberg atoms, the thing is that they are so large, right, that they are easily analyzable. You can analyze the signals much easier, and the signals that you get are much stronger. So that's the reason. I mean, you measure magnetic fields with rubidium vapor in room temperature in just normal hyperfine transitions, and that's very, very sensitive. But if you want to do it with that sort of sensitivity, you have to go to Rydberg atoms.
Okay, so I think there was a question back there on the table, and then here, and then here.
Wow.
So
That's
it's
a
about
lot.
rotating particles. Just wondering whether you try to tether these particles on a protein. For example, there are proteins which do circular or like rotating motions. So whether you can tether these particles on the protein and try to enhance their catalytic activity.
No.
Or for example, on bacteria, which has a flagella, which is also rotating to move the bacteria, then you can sort of like make them guided.
So no, I have not done that. I have to say that I didn't know that there were proteins which will do this, but I'm learning slowly, but I am. And as far as bacteria with flagella are concerned, we're doing other measurements with them, but not for rotational dynamics. Because the reason for it is that I can spin them, my baterites, as fast as I can, as I want, without doing anything else with them. I just zap them with more light, and they will spin much faster. So as I said, I can go to megahertz, high megahertz, and I can spin it. Now, the question that can be asked is whether, if I use it in biological systems, whether it introduces some thermal effects that will destroy my system. And we have tried to do measurements and show that it does not cause big problems because we are using 1064 laser, which in this particular birefringent material is not absorbed very much. But the question of swimmers, like bacteria with flagella, is super interesting, and we have done a lot of work on that. I can tell you more about it, especially looking at active matter.
Thanks.
Thanks for the presentation. I want to ask some more questions about the active microbiology. In particular, if you rotate the beads at such high shear rates that you have shown, aren't you worried that you leave the linear viscoelastic regime and then you start measuring nonlinear effects that are not well
I can
captured?
measure viscoelasticity. I can measure both G prime, G double prime.
Probably leave the linear viscoelastic regime.
Well, as I said, I can deal with viscoelastic regime, but I never measure viscoelasticity or viscosity with the particles which are rotated very fast.
Okay, so you are trying to stay within the linear regime of the material response.
Yeah,
okay. And the other question I had about this is, what is the vision or like the aim that you try to achieve with such rotational measurements? I mean, with active microbiology, you would usually probe what is the non-equilibrium property of the biological material, so on and so forth, but what would be the aim for rotational?
It's the same thing. It's just we believe that with rotational geometry, we can reach to the systems, first of all, to much smaller systems, volume-wise, and also looking at non-equilibrium systems. So that's the dream. And especially with the inertia measurements, which show that I can go to very short time scales, follow the non-equilibrium process from the start to long times. That's the dream.
Thanks.
Thank you very much for a nice talk. Regarding to the dumbbell trap, the atomtronic circuit, I wonder how long this circuit can stay, because I assume when you release the BC and it starts to oscillate between two reservoirs, then during all of this oscillation, the atoms start to heat up and some of them get out of the trap very quickly. So therefore, it cannot last for a long time. Is that correct?
So there was a curve which was showing you the Josephson oscillations, and it's in a certain time, and after that it decays. You are absolutely right. But for that time period, I can measure it. So it's limited, but it's measurable. And as I said, what is the most important is that the reproducibility of creating those pictures from one sort of BC till next is very, very large, very good. So you can do it, yeah.
Any more questions from the audience? There's a very general question from online, if you are able to detect cancer cells.
Cancer cells? No.
Okay.
Very short. Very short answer.
Other questions now? If not, then we will also have cake and coffee outside, so you can also talk.